Germ layer and fat mapping techniques. In the year 1817, Christian Pander, a doctoral student at the University of Würzburg, Germany, first recognized the existence of germ layers in chicks, that is gallus. So what is germ layer? A germ layer is a group of cells in an embryo that interact with each other and contribute to the formation of all tissues and organs. Development of the germ layer begins at the later stage of fertilization, wherein the formation of zygote takes place as a result of fusion of male and female gametes, that is spermatocytes and oocytes respectively. The zygote then undergoes subsequent cleavage by mitotic division, wherein one cell is divided into 2, 4, 8 and so on and transforms into a hollow ball of cells called blastula. The blastula then undergoes an important process called gastrulation, wherein the formation of germ layers such as ectoderm, mesoderm and endoderm take place. Together, the three germ layers will give rise to all the organs in the body. Based on the presence of number of germ layers, animals are classified into the diploblastic and trypoblastic animals. A diploblastic organism has only two primary germ layers, namely ectoderm and endoderm, whereas a trypoblastic organism has an additional layer called mesoderm. Now let us understand formation of zygote by fertilization. Fertilization is a process in which the fusion of male and female gametes result in the formation of zygote. In human, after about the third day of the fertilization, the single-celled zygote undergoes multiple cleavage by mitotic cell division to form a solid mass of cells called morula. So what is morula? The morula is then transformed into the blastocytes consisting of an outer layer called as a trophoblast and an inner mass called as a embryoblast. The embryoblast initially has a two layer, namely the hypoblast and epiblast. At the end of the second week, a primitive streak appears and the epiblast in this region moves towards the primitive streak, jumps down into it and forms a new layer called the endoderm, pushing the hypoblast towards the exterior. The epiblast keeps on moving and forms a second layer called as a mesoderm, and the top layer is considered as the ectoderm. So what is gastrulation? This gastrulation is the embryologic stage of germ layer formations. During earlier embryonic development, the overall architecture of the body and the major axes are established through the process called gastrulations. These, in fact, in its original term, refer to the formation of a gastric cavity, the archenteron. However, today the term gastrulation is often used as a synonyms of endoderm and mesoderm formation. During gastrulation, a hollow cluster of cells called a blastula recognize into two primary germ layers, an inner layer called endoderm and outer layer called ectoderm. In all vertebrates, these progenitor cells differentiate into all adult tissues and organs. Gastrulation is a dynamic process in which cell migration and epithelial to mesenchymal transitions occurs in an organized way by switching the cell adhesion molecules. The gastrulation step begins with the extensive cell mixing followed by migration of mesenchymal cells to the presumptive middle line to form the primitive streak. It is the site at which epiblast cells undergo EMT and initiate the formation of distinct cell layers that will be form the primary germ layers mesoderm and endoderm. Epiblast cell that do not enter EMT undergoes differentiation to become ectoderm. Several transcriptional programs that influence cell migration during gastrulation, and subsequent cell differentiation into the three germ layers are activated by intracellular signaling mediated by WANT, FGF and TGF beta ligands. In most animals, endoderm formations goes parallel with the formation of third germ layers, the mesoderms. Now let us talk about development of germ layers, organs and tissues. So what is the endoderm? In general, the blastocytes grows in size by obtaining nutrition from the uterus, and a few cells separate from the inner mass to form endoderms in blastocoels. The cells migrating inward along the archenteron form the inner layer of the gastrula, which develops into the endoderm. The endoderm then differentiates into the primitive guts. A part of it gives rise to digestive tract and the other portion forms yolk. After the formation of endoderm, the remaining cell of inner mass forms embryonic disc, which has three parts, namely cephalic margin, embryonic disc and caudal margin. In the beginning, the endoderm consists of flattened cells that subsequently become columnar. It forms the epithelial lining of the wall of the digestive tract except part of the mouth and pharynx and the terminal part of the rectum. It also forms the lining cells of the gland which open into the alimentary canal. Together, the endoderm forms the epithelial parts of trachea, the lungs, the pharynx, the thyroid, the parathyroid, the liver, the pancreas, the stomach, the colon, the intestine and the urinary bladder. Now let us talk about the mesoderm. The formation of mesoderm in the embryos is the characteristic feature of triploblastic animals. During the gastrulation, some of the cells migrating inward contribute to the mesoderm, an additional layer between the endoderm and the ectoderm. The formation of mesoderm leads to the development of the coelom. The coelom that fills with the fluid acts as a cushion and protect the organs that formed inside the coelom from shocks. Thus, the organs in the coelom have the freedom to move, grow on its own pace and develop independently of the body wall. The mesoderm has four different components such as intermediate, paraxial, lateral plate and chorda mesoderm. So, the mesodermal cells begin the process of differentiation through interaction with the ectodermal and endodermal cells using different cell signaling cascade mechanisms. The intermediate mesoderm gives rise to kidneys and gonads. The paraxial mesoderm forms into cartilage, skeletal muscles and dermats. The lateral plate mesoderm is responsible for the development of the circulatory systems, the wall of the gut and the wall of the human body. And finally, chorda mesoderm form the notochord. In brief, the mesoderm form muscles, bones, cartilage, connective tissues, adipose tissues, circulatory system, lymphatic system, dermis, genitourinary system, serous membrane and notochord. So now let us talk about ectoderm. The embryo's epiblast gives rise to the formation of the ectoderm, which forms the outer layer of the embryo. The ectoderm develops into the surface ectoderm, neural crest and the neural tubes. The surface ectoderm develops in epidermis, hair, nails, lens of the eye, sebaceous glands, cornea, tooth enamel and the epithelial of the mouth and nose. The neural crest, which derived from the ectoderm, is often considered as a fourth germ layer owing to its great importance in the development of peripheral nerve systems, adrenal medulla, melanocytes, facial cartilage, dentin of teeth. The neural tube of the ectoderm also involved in developments into brain, spinal cord, posterior pituitary gland, motor neurons and retina. Now let us see fat mapping, the development of an organism from a single cells, crossing through different stages of embryo, morula, blastula to a recognizable physical form that slowly shapes into a small animals is an orchestrated event of different mechanism that occurs at a time bound harmonized way. It is indeed a miracle to not only a common man but also to the developmental biologist, who astonished by the finely tuned developmental process termed morphogenesis. It is simple to ask as how this process is finely tuned and what controls the regulation. Classic way to approach such a question is to generate a fat map. This area of developmental biology is the recent focus, and developmental biologists have carefully characterized the stage of morphogenesis and have identified key transitional periods, blastula, gastrula and neural stages through varied imaging methods collectively called fat mapping. A fat map is a graphic representation of the cells within the embryos that are developing into specific tissues or organs at a given specific point during developmental stages. For example, studying the future position of a dividing cell from its origin from a mass of cells, that is cell lineage, or studying the developmental differentiation of a particular cell forward from its position in one of the three germ layers. Labeling individual cells within their germ layers will allow for a pictorial interpretation of gastrulation. This chart of graphical representation that explain the fate of each part of an early embryo is referred to as a fate map. Constructing a fate map is an important part of understanding the developmental pathway of an organism. Determining the architectural assembly of an organism could possibly lead to establish the function of each specific region. Furthermore, understanding the lineage and migration of progenitor cells, which help in discovering gene regulatory networks and signaling pathways. The possibility of new developmental discoveries come with the creation of each new fate map. Now let us see construction of a fate map. A fate map is an experimental tool to explore the recognition of the cell from one stage of development to a later stage. Briefly, this step involves labeling of a cells at a recorded location and observing the same cell or its progeny that are relocated at the later stage. If cell rearrangements are conventional from embryo to embryo, then a map can be constructed by repeating this procedure several times from different recorded positions. This approach has been widely used, employing a variety of ways to label the cells and varying degree of complexity in classifying final cell fate, ranging from scoring their location by tissues or organ or analysis of gene expression patterns. In recent times, various techniques have been devised from the construction of the fate map. Of these, tracking the course with natural colors and artificial markings are most important. Now let us talk about natural markings. Fate mapping can be performed utilizing the natural markings or pigments that are present in an organism. For example, the cytoplasm of certain eggs of ascidians has a natural pigments. Thus, in the fertilized egg, 4 colored centers can be seen as an upper hemisphere of tight protoplasm, a yellow crescent posterior ventrally, a grey crescent anterodorsally, and a vegetal area of dark grey yolky substance. The fate of this area can be followed very easily and has been revealed that the upper clear cytoplasm contain the material for epidermal ectoderm. The grey crescent are differentiated into the prospective neuroectoderm and notochord. The yellow crescent becomes the prospective mesoderm and the dark grey yolk area forms the prospective endoderm. Now let us talk about artificial markings. There are three methods to mark or label only the blastomere by which their fate can be traced. Now let us talk about vital staining. This vital staining method was first introduced by Vogt in the year 1928. Vital stains such as methylene blue, neutral red, Bismarck brown, Janus green and Nile blue sulfate are some of the common vital stains which are retained by living cells for considerable period of time. These dye are in no way found to interfere with normal life activities. Thus, the persistence of the stain in the cells make it possible to determine the ultimate location of the marked area in the late embryo. So, the vital staining technique involves soaking a piece of agar with a vital dye and placing it on a surface of the embryo in the required location. Upon exposed to tissue, the stain immediately diffuses into the blastomeres and the cells are stained. The interesting feature is that the descendants of the marked cells also become stained. Therefore, in the differentiated embryo, the part stain indicates the fates of the original stained blastomeres. For examples, if the stained cell are seen in the gut of the differentiated embryo, the fate of the marked blastomere is said to be the developmental of gut. Similarly, several such stain marks can be made on the surface of the same embryo using different color and the various organ forming area can be marked in this way. Now let us talk about second method, carbon particle marking. This technique was introduced by Spratt to demonstrate the process involved in primitive streak formation in chick. This method consists of applying tiny particles of carbon over the surface of blastomeres which stick to the cell surface and enable the investigator to monitor the movement of the cells and to determine the fate of these blastomeres. Now let us talk about third method, radioactive isotope labeling. Radioactive isotopes have long been used in the tracer experiments utilizing the property of emission of alpha, beta and gamma rays. Briefly, the earlier blastomeres are labeled with any of the radioactive isotopes such as C14 and P32 and cores of these radioactive isotopes can be carefully monitored to determine the fate of blastomeres. To summarize, at the end of this lecture, we understood the germ layer has main role in the formation of tissues and organs. They form the main body parts in the animals without germ layers. It may be difficult for formation of body organs. For the help of the fate map technique, we can understand the future change in the germ layers of tissues and organs in animals. Thank you.